Good morning, everyone. Thank you for joining today's distinguished lecture organized by the Faculty of Science, the University of Hong Kong. Today, we invite Professor Hongyu ZHAO to give a lecture. Let me pass the floor to the Dean of the Faculty of Science, Professor Matthew Evans, for introduction to the speaker. Thank you, Matthew.
Okay, thank you very much, Michael. Dear teachers, students and guests, welcome to the Faculty of Science Distinguished Lecture entitled Characterizing the Genetic Basis of Complex Traits through Integrated Statistical Modeling and Analyses. I'm Matthew Evans and I'm the Dean of Science. Although genome-wide association studies, GWAS, have been a great success in the last 15 years, in identifying genomic reasons associated with complex diseases and traits, major challenges remain both for identifying functional genes and variants in statistically significant regions, as well as interpreting the GWAS results. These challenges are due to the low signal-to-noise ratios in GWAS analysis, complex dependence among genetic markers, and the lack of access to individual level data for many studies. In this lecture, Professor Hongyu ZHAO will introduce a statistical model that is commonly used to characterize the genetic contributions to complex traits and its robustness to model misspecifications, as well as extensions to identify relevant tissues or cell types for a specific trait. He will also discuss statistical inference using either individual genotype and phenotype data, a typical setup for traditional statistical analysis, or summary statistics, which are more easily accessible from GWAS. It is my great pleasure to introduce our speaker, Professor Hongyu ZHAO, today. Professor ZHAO is Ira V. Hiscock Professor and Chair of Biostatistics at Yale University. He received his bachelor's degree in probability and statistics from Peking University in 1990 and a PhD in statistics from UC Berkeley in 1995. His research interests are the developments and applications of statistical models in molecular biology, genetics, drug developments, and precision medicine. He received several honors, including the Mortimer Spiegelman Award for a top statistician in health statistics by the American Public Health Association and the Pao Lu Hu Prize by the International Chinese Statistical Association. His research has been recognized by the Evelyn Fix Memorial Medal and citation by UC Berkeley, a Basil O'Connor Starter Scholar Award by the March of Dimes Foundation, and he is also a fellow of the American Association for the Advancement of Science, the American Statistical Association, and the Institute of Mathematical Statistics. So without further ado, I will now pass the floor to Professor ZHAO. Thank you.
Okay, thank you so much, Dean Evans, for a very generous introduction. Also, thank you, Zhonghua and Michael, for inviting me, and also thank a lot of friends to show up. And I'll just maybe share my screen. I'll start. Okay, can you see my screen?
Yes.
Okay. All right. So today's talk is really about the work we have been doing in the last how many years, including a lot of contributions from people in the audience like Chan. Actually, he has contributed quite a bit to what I'm going to talk about today. So I'll just walk you through more about the like stories on how we use genetics and these different tools to understand the basic genetic basis for complex traits. And they all started out more than 100 years ago. And this is a very young, like a picture of young R.A. Fisher when he was in his college time, that he wrote this really probably one of the most seminal papers in statistics that laid the foundation for the whole field of quantitative genetics. So as says the title of the paper is really the correlation between relatives on the supposition of Mendelian inheritance. And in this paper, there's a lot of like novel concepts introduced, which now become like common house names, variance, ANOVA, assortative mating. So this is really having to do with how we really understand the genetic contributions to a lot of traits. For example, height. I show here the Ming Yao, his wife, and their young daughter. And even just a couple of days ago, I was reading the news and talking about how tall she is. She's been growing, and how tall she will grow into. There's still we can still take a bet, right? That's Ming is like 2 meters 26 centimeters, and his wife is 190, and their daughter may well grow up to 2 meters. Yeah. So, but what's the basis for this? And because this is all even back before R.A. Fisher, the Francis Galton, and by collecting the data from the parents and the children, they look at joint distribution for height, and really say, okay, there's a lot of similarity between parental heights and their children's height. There's also correlation and regression. So there's a lot of interesting things that were very, very new at that time, but now it's just already in our blood to think about genetic, the correlation, etc. Right? So this really the understanding or the phenomenon of genetics has really driven the field of statistics from the very beginning. Okay. Just to lay out what Fisher was thinking at that time about how to use genetics to explain the heritability or the trait of the passing from parents to offspring. So we have to come up with some mathematical models, right? So it's easier just to think about this in the quality terms, but it's much more convenient or to get some insight to write the equation. So this equation is not difficult. And for the most part of my talk, the equation will be like this, right? So it's nothing more than this additive like linear model to talk about. So let's all this think about this scenario about, for example, look at height. We take the response variable is a height. And for any like a step 101, say, let's regress this white onto some so-called predictors, explanatory variables, right? So what are explanatory variables here? For height, and you think, okay, there must be some genes, right? Some genetic factors. Let's call it G. And then there may be some environmental factors like the nutrition, exercise, etc. And then there's some noise. Yeah, so this is a very simple model to describe how the height may be determined or affected by the combined effects from genes and environment and something else, right? So talking about genes, and then we think like, I think all of you know, like we have 2 sets of chromosomes in your body, right? So there's a parental side, there's a maternal side. And then we can really I think about there's a 2 sets of chromosomes, therefore there's a genotype. So genotype has 2 letters, right? One letter corresponds to what you get from your father. The other letter corresponds to what you get from your mother, right? So for example, there's one major gene determines someone's height, and that gene has 2 different forms in the population. There's a like tall gene or short gene, very simple way. So we call the tall gene Q and the big Q, and the short gene the little q, right? So there's 2 different forms of this particular this gene in the population. If you're lucky, you get 2 big Qs, you're tall. If you're not lucky, you get 2 short like genes, like we call the alleles here, little q, you're short. So this is a very simple model. So in the population, there are 3 different genotypes. You can get 2 copies of big Q, 1 copy big Q, or 0 copies of big Q, so 2 QQs. Right, so there's 3 different genotypes in the population. So this is all I think about like now, because you get 2 copies, 1 copy from your father, 1 copy from your mother. And if you have this, the big QQ, the tall genotype, assume these people have average height A. And you get little QQ and minus A to make it a little bit more symmetric, right? So there's a population average that we'll make sure they add up to be positive. So there's no negative height. It also makes things easier, right? So we assume people with a 1 big Q, 1 little q is sitting somewhere in the middle, right? Between A and minus A, that's 0. So it's as simple as it can be. And also, not only do we know about there's a big Q, little q, and also we say out of the population, there may be like a 70% people with a big Q, or 70% of the genes have a big Q, and the 30% maybe little q. And we assume the 2 copies you get, they are independent of each other. So the big QQ is like F squared. So F is a frequency of Q or proportion of Q. So this is called Hardy-Weinberg equilibrium, right? So there's a particular name for this independence assumption. So now actually we have a model. Let's ignore the E here, say all the height, right, really depends on the genotype, big QQ, big Q, and little q, little q. And in the population now, we know there's F squared people with big QQ, 2F minus F with a big Q, little q. So this is a very well-defined model now. So now we can start from this model to really see what we can learn about the genetic contribution to a trait. So how to quantify or how to rationalize the genetic contribution to a trait. So this is look at the whole overall population here, right? So this is the people with big QQ, big Q, and little QQ. There's a mean, there's certain spread, some noise, and this is a mixture distribution across the whole population here, right? So this is the model again. And now we can really look at the variation of height in the population. We can partition that variation into the contribution from the genes, contribution from like environmental factors and noise, right? So we can write this, the variance. Now talk about the analysis of variance. The variance of the height, you can, it's a very simple additive model. We say that it's additive effects from the variance due to genetic factors, variance due to environmental factors, and something else, right? Yeah, if you assume this very simple additive model, d is equal to 0, if the Vj and Va are the same, right? So now you can quantify the genetic contribution to the trait by taking the ratio of Va over Vy. It's a proportion of the variance of height that can be explained by the contribution from the additive effects or genetic effects, right, for the trait. So therefore, we can estimate the we have a way to define the heritability, right? So for height, we always say, okay, if the parents are tall, the kids are tall, and but is any way to really quantify the degree of heritability, right? So, so back more than 100 years ago, people didn't even know how many chromosomes in the human, like in the human cells, and let alone what these things were. So R.A. Fisher went to a different way to really estimate the heritability defined through this term. So how to do that? So he went to resort to relatives, right? As Francis Galton did, right? Look at the parents, look at the offspring. We can look at the resemblance, right, between relatives. For example, this is represent the height for father, the height for the son, right? So we have a lot of fathers, a lot of sons. Now we can calculate the covariance, like how similar the son's height is to the father's height. And the covariance here, it turns out, has a very neat form. It's essentially equal to CR is a coefficient that quantifies the genetic relationship between the two relatives. For example, between father and son, the CR here is 1/2, right? That says the covariance between father and son essentially is equal to half of the genetic contribution to the height. So therefore, if you observe a lot of like a father-son pairs, you can estimate the covariance. You multiply that covariance by 2, you recover the genetic contribution to height, and then you'll be able to estimate on that contribution. So this is all great, right? So this is under a model when there's one major gene contributing to the overall height phenotype. And then it turned out at that time, people realized, R. Fisher realized, there's no such a major gene, right? For height, it's really determined not by one gene, but probably by the additive effects of many, many different genes. So why is that? If you assume there's like a major genes, for example, there's a tall gene or the short gene, and suppose each one has 50%, right? So there's 25% tall, 50% tall, short, 25% short. You will see this very discrete type. I mean, but in reality, we don't always use the height as a normal distribution. So if you just assume there's like a major genes, the mixture them together will not look like normal, right? So the way that R.A. Fisher like thought about this problem said, so let's instead of assume there's one major gene, let's assume the height is due to the additive effects of many genes, right? So this is why instead of being the additive effect, one major gene G plus E, not big E and little e, is due to the additive effects of many genes, right? G1, G2, G3, M can be very large, and you can collect all these different genes, additive genes, into one, like a bold-faced G. So now this big G, the bold-faced G, is no longer one gene, but additive effects from many genes. But it turns out mathematically speaking, under this model where the nature is very different now, there's many genes contribute to the height, but the covariance, the form of the covariance between the parental height and child's height still retains this very simple form, right? The covariance between the two relatives, Y1 and Y2, is still correspond to the degree of relatedness and the genetic contribution to the trait, right? So this is really great, right? So if you believe this model is right, you can collect a lot of different kinds of relative pairs. You can estimate their covariance, and you are still in a position to estimate the genetic contribution to a trait, right? So you don't need to know precisely what the genes are, as long as you believe this model is a good representation or approximation to what's going on in biology, then you're in a position to make some statement, right? So based on this, you can use this so-called pedigrees, having a lot of related individuals, you can estimate. So these people have done this for like more than a century. Like height, people estimate maybe 80% is due to genetic. Body mass index, which is defined by the body weight in kilogram divided by the square of height in meters, right? So this is a 0.45 to 0.8. So that's a range of heritability. And also if you look at even for like some other like psychiatric traits, like neuroticism, like somewhere between 13% and 58%, different studies have different estimate. But regardless, there's a significant genetic contribution to these traits, right? From the study of relatives. We are able to estimate because we have a model in place, and this model allow us to derive the relationship between the resemblance between relatives and the overall contribution of genetic factors to a trait. So this is very neat, right? So again, just to summarize what I talked about in the last 15 minutes, it's a very simple RE-Fisher model proposed like more than 100 years ago, like relating the height or a phenotype, quantitative phenotype to the additive effects of many genes. And based on that model, actually, we can estimate heritability without knowing which genes contribute to a trait if you assume the model is correct. So that was the model established in 1918. Between 1980 and 1953, people didn't even know what the genetic materials were pretty much. And 1953 wasn't quick, right? It's the proposal of the DNA. And then in the 1950s, people realized there's 46 chromosomes in human, there's 23 pairs. And in 2001, people have the sort of initial sequencing of the human genome. If you follow, or if you like the literature or there's some like pub media, recently they only after this year is the 20th anniversary of the first publication of the human genome. And also this year, people really have the first end-to-end human complete genome sequence, right? There's still, people are still trying to do many different things. But back in 2001, we have some idea about what the genome looks like, right? So there's a big improvement from 1918 to 2001. So not only we know how the genomes look like across the population as more like consensus, but also people start to really understand the complexity also as the variations across different genomes. That's why people are different, right? So this one very common form variation people see is called the single nucleotide polymorphism, right? So this is a 5 nucleotides here, 5 nucleotides here, and the 4 nucleotides are the same. And these 2 chromosomes differ as 1 nucleotide. This is called single nucleotide polymorphism. So there's variations in the genome corresponding to what I said, the big Q, little q. You have this C, you could maybe tall, the G maybe short. But there's a lot of variations in the human genome, right? People know about tens of millions of such SNPs in the human genome across different populations. Not only people know about what these things are, and also people have a way to measure them, right? So this is one of the 2 common platforms people use to really measure, observe the genetic variations called the microarray genotyping arrays. And so this is the Omni 1 quad, and that's a microarray that can measure maybe 750,000 or 800,000 SNPs in the genome. So this is people can really estimate those, measure them at a very large scale, right? So there's a lot has happened since 2001, and people already have this platform in place. And so with this very powerful instrument and the knowledge, now we can really ask the question, what genes, what variants really contribute to different phenotypes, right? But remember, back to Fisher time, we can conjecture. So there's such variation, and based on arguments, on reasoning, we have mathematical equations. We can look at relatives. We can estimate heritability, right? But now actually we are in a position to look what these genes are, what the variations are, and to see what genes, what the variations are really associated with different phenotypes. So one way to do this through the so-called genome-wide association studies. And by the term genome-wide association means look at all the markers in the genome, trying to do association studies, right? So you collect thousands up to hundreds of thousands of subjects, and I'll show some of the numbers. And you do these genome-wide markers, right? So the one I show you before, there's a few 100,000 or a few million markers. And then you look at association between these markers and the phenotypes, and to really see what markers are associated with what phenotypes. Just in terms of scale, people are really analyzing the data, the size, the UK Biobank, about more than 500,000 people. Every single one has been genotyped, right, for 700,000 markers. And the US median veteran program in the US, and the goal is to recruit 1 million people. We already got more than 850,000 people. All of us is initiative from the US. The goal is 1 million people. The FinGene Finland is a small country, but they already got 460,000 people. On the China Kuduri Bank, Kuduri, I think I heard is a billionaire in Hong Kong, based in Hong Kong, but he donated a lot of money to do research, and this sort of the bank, with his support, built 500,000 people. Biobank Japan, 200,000 people, and Taiwan Precision Medicine Biobank, the goal is to recruit like 1 million people, probably more. So a good thing about the median veteran program, the FinGene and the Taiwan Precision Biobanks, these few several biobank projects, they are also have coupled extensive electronic records, not only just observe height or blood pressure. You observe pretty much everything. These people go to the hospital, go to healthcare, all the things are entered in the database, and you actually look up all sorts of things, the phenotypes. Okay, so this sort of data we are analyzing and these days, and we can look at, maybe pick out one trait, look at the association between this trait and the many different markers. You can make adjustment for sex, age, etc. Right? So with all these the big data and also a lot of discoveries, and so one question people can ask, we all know we are no longer happy or satisfied with knowing height is a genetic trait, but rather we want to know, can we really predict someone's height based on his or her genotypes, based on what we know about the genetic makeup or someone. So you can also formulate to different questions. How much phenotype variation can be explained, right, by what you can identify from this identified variants associated with different traits. Yeah, so this is a question people actually start asking even by the early days of GWAS. The first GWAS study was published in Science in 2005 by our colleague in our school, and Josephine Ho, like looking at age-related macular degeneration. So this is actually not too long after that, about 2008 or 3 years after that, because of height is such common trait, right? Everyone has a height and also can be measured with a pretty good accuracy. And you can easily design a study with a lot of people measure for their height, as long as you get their genotypes. So this is one actually marked by Chan, saying like 2008. And this is a study looking at tens of thousands of people from Iceland, the Netherlands, and other countries. They identified 27 regions in the genome associated with height. And then you ask the question like, now we know height is a highly genetic trait, and how much we can predict someone's height based on 27 regions, right? This marker from 27 regions, lo and behold, like 4%, right? So as showed you earlier, the heritability estimate from studied families, relatives, about 80%. So this is a little bit disappointing, like the 4% versus 80%. So maybe the study, the sample size was too small. And so fast forward a few years, 6 years, this study with 250,000 people, instead of 27, identify about 700 variants. If you also going beyond 700, you include the top 2,000, 4,000, or 10,000, you can get a model to predict someone's height. Yeah, you can see how accurate that model is, like 21%, 24%, 30%. That's still a big way from 80% with the data from the result from 250,000 people. Yeah, now 700,000 people, they identify even more And the number here 25%, right? So there say that we know study after study from the study of the relatives, we estimate the heritability for height about 80%. From the data I just showed you, anywhere from 4%, 10%, 24% there. So there's a big difference between 24%, 80%. So what's going on? And in the late 2010s, early the late like the 2000 early 2010s, people thought maybe there's deficiency in the platform because we are using microarrays, right? We are only capturing the common variants. Maybe the rare variants explain a lot of phenotypes. So that's really technology deficiency. We are not really we are missing a big part of the whole genome, right? So the other one, really deficiency in the models, right? You may say, this is the model too simple. While the RA Fisher is more than 100-year-old model, there's only very simple additive model. There's no interactions, there's no nonlinear effects. And so that probably that's the reason why you don't get good fit. But then came out this paper on 2010, that's 2 years after the other paper, like 4% paper. Actually, this one caught a lot of people in the field, including myself, by surprise. Yeah, so in this paper, with only 4,000 people, remember the paper where only 4% of the variance explained by the genes or found, based on tens of thousands of people. This is only 4,000 people, but they made a statement that 45% of variance can be explained by considering SNPs, all SNPs simultaneously. So that's a pretty big statement. So we'll basically say that there's probably is not going to be 25% or 30%, actually 45% of variance may be explained by the SNPs simultaneously on that microarray. But how to reconcile this big difference here? So this paper is answering somewhat different questions. The question is, how much heritability can be potentially explained by all the study SNPs and the potential effect size? Is this possible, right? This paper says it's possible. And so let's talk about their model and to see the connection with RA Fisher's initial 1918 model and also the statistical tools that they used. So again, in this paper, the model, like the RA Fisher, if I look at RA Fisher's model, we assume the Y, let's forget about the environmental factors, is additive effects a lot of different genes. That's what RA Fisher has, right? That's what, and then he proposed to use the relatives to estimate heritability. But now with all the data observed from genotypes, we know all different genes and also their variants. Actually, not only we can put in more abstract term, very abstract, but we can make it a little bit more concrete, right? So now we can really say Y is equal to the additive effects, a lot of different variants in the genome. And for these microarrays or the genotyping arrays, we are able to estimate or observe the genotypes, the genotype score, like C1, C2, C3, right? And if you have a very good estimate, the effect size of each SNP, so this is the model we can use to predict someone's height, right? So this is a prediction model. The height is an additive effect, a lot of different genes. We know what the different the genetic variations are. This is the score, and also we know the effect size, right? So this is really one. And also, if the initial GWAS, what people do is to get all these genotypes, you estimate effect size. That's a model, okay? But the thing is, it's very challenging to infer you. And even when we have a large sample size, it turns out there's maybe thousands of genes associated with the height. And each gene, each variant only has a very, very small effect. So the estimation here is going to be very challenging, right? So even if you have a very good observation of the genotype, the yield here can be problematic. And that's why the estimation is only the R square only hold around like 20%, 30% in that range. That's the challenging part. But in statistics, there's a different way to think about this setup. Is saying instead of thinking about all these U's, right? U1, U2, U3 as a so-called fixed effects. We want to really estimate the effect size of this U, these different SNPs on Y. But let's think about a different model called the random effects model, right? The random effects model says, let's assume for those different genes, the effect size, all the effect size come from a common distribution. Let's assume that distribution to be normal. with mean 0, and the variance sigma g squared over m. So sigma g, you can think about the overall contribution to the overall genetic contribution to the trait. Let's assume every single variant here has equal opportunity, has equal share of contribution to the overall genetic contribution. So let's assume all the U follow the common distribution with a 0 mean, and the variance sigma g squared over m. M is the total number of markers, right? Suppose you're looking at 1 million markers, assume this is to be sigma g over 1 million here, right? So this is a very simple model. But good thing about this simple model is that now instead of estimating every individual U and trying to come up with some way to predict, actually you can estimate sigma g squared from this model using the data you have. So essentially the idea is you can look at the phenotype similarity to the genotype similarity across all the markers. And so this is the, you can actually calculate the covariance, because for Fisher's model, the covariance between relatives, right? Then the derivatives are defined by their degrees relatedness, like the first degree relative, second degree relatives. You can think of this more as another form to write the covariance between relatives as a covariance between two individuals as a function of their genetic similarity, C transpose. You observe the genetic similarity between two individuals, and this gives away more a counterpart what our Fisher had for relatives. But now you use observed genetic markers to quantify the similarity. And under this so-called random effects model, and there's a well-established method called REML, right? You can use the restricted maximum likelihood estimate. And so that's the basic idea what's in the paper. So again, to recover what I said, the Fisher's model allows estimate heritability from family data, but random effects model allows you to estimate heritability sort of the so-called the chip base, or the heritability can be explained by the markers on the chip by correlating the similarity between two individuals with the similarity between these two individuals across all the markers. So you use all the genome markers to quantify or measure their genetic similarity. The same idea, but in different contexts. Okay. Yeah, and now if back to this table we saw before, this is for the family-based heritability estimate, 80% for height. If you use a GWAS HIS, you do the fixed effects model. So at this time, maybe 10%, now it's 30%. But if you use the random effects model, that's 45%. Okay. So this is all good, right? So it really tells you that under this model, if you believe that this model is right, it's not that the platform is inadequate. It's not because the model is too simple, but rather just because we don't have enough sample size to really precisely estimate all the effect size for all the markers. So this is the major challenge. And so that really tells a good way, statistical models, I'm thinking, can be quite powerful to really look at what's in the data. But the problem is the random effects model makes a very strong assumption about that every single marker has equal contribution to the trait, which basically says everywhere in the genome contributes to a given trait, which clearly is probably not realistic. Yeah, so this random effects model is clearly misspecified, right? So instead of having every single marker contribute to the trait, maybe only a small proportion of the markers contribute to the trait, right? And but the REML, the whole machinery assumes every single marker works, right? So the question, how much we can really trust the results in such when the model is specified. So this is sort of the exercise. Actually, this is the data when Chan was at Yale, we're trying to play with. And so let's assume the model that all the markers contribute to the trait. The true heritability is 60%. This is a simulation, right? If every single marker contributes, and the REML model assumption holds, and we recover 60%, right? But now let's assume the model is specified, and only 50% contribute. Now we still get pretty unbiased estimate. And we can keep doing that all the way until there's only 10 markers of 20,000 contribute to trait. REML, this well-established statistical method, still works. To us, was really big surprise. And actually, we reached out to two colleagues, UC Davis, Jiming Jiang, and Dabashis Pao. And actually, it turns out there's a mathematical reason that why this still works. It's really just to show that there's some really deep, some reasoning why the traditional REML was designed for like standard random effects model. When the model is specified, yeah, it's still very robust. This is a very neat. And so one thing that we assume in that paper is assume all the markers are independent. But in reality, these markers, these SNPs, they are not independent because when the markers nearby, so this just shows you there's 3 different populations of white, East Asians, and African population, and the red here means a strong dependence, right? So this says these markers are not independent in the human genome, and whereas our model assumption, they assume to be independent. But again, when we look at the real data, and we can simulate from real data, when we assume the markers are dependent, the REML is still pretty robust, right? So that's very good to show that there's still something that mathematically still needs something that can be done there to prove the robustness. So the other thing that REML, the whole setup requires individual level data, which means that we assume we have both the phenotype observed for everyone as well as the individual genotypes. But in genome-wide association setting, due to privacy issues or other political considerations, people are not willing or not able to share individual level data, but rather much easier to get the summary data. So what does summary data look like? So instead of having the very large rectangle table with individual phenotypes and all the marker genotypes, what we have here is the association statistics for each marker, right? So this is the summary statistics. So this marker 1, marker 2, marker 3, you know the alleles, different forms, allele frequency, the effect size, standard error, p-value, and the number of individuals that we have sort of information about that marker. Okay, and so LD score regression was introduced by people in Boston a few years ago, and that the LD score regression base says that established the relationship between you can think about the marginal summary statistics and the degree of sort of something to do with the heritability. And also not only that, but also called the LD score regression, because the observed statistics, marginal statistics, not only has to do with how well on average each marker contributes to the trait, but also depends on how dependent this marker is to the other markers in the genome, therefore called the LD score regression. So in the interest of time, I will now go through this, but just saying that this LD score regression actually is a very powerful method. Also, it's computationally much more efficient to do. And also, when the markers are independent, the REML is to really use individual level data. LD score regression based on like the marginal, and it just show the different method comparison in terms of the efficiency. So this is the REML method. This is the LD score regression. There's another method, but pretty much you'll see that there isn't too much of lost information there by going from individual level data onto summary stat by looking at the spread here. But on the other hand, if you have the marker dependence there, you do see there's can be quite a bit of lost information. And people also have been making some effort in recent years to recover as much information from the summary stat. So that's a different topic. More recently, and also in collaboration with the Jiming and Dabishes, also we're trying to extend what we proved in the earlier paper on the individual level data on the summary statistics, also to show the robustness, both for the heritability estimate and also as well as a covariance estimate I'm going to talk about next. Okay, so that's essentially the first part of my presentation, that to show this is what people have done and what we have done, and the power of statistical thinking approach, and both for individual level data as well for summary level data. So the nice thing, the segment is going to inject a little bit more biology into my presentation, saying, and so far we have been treating every single marker as random variable, but in reality, these markers, they are not just like equal. Some markers are more important than others, because if you think about this, the whole human genome, just give you a very simple illustration here, and in the whole human genome, about 3 billion base pairs, but only 1.5% are in the coding regions. Only like 30, 40, 50 million base pairs they're coding. So therefore, when a marker in the gene in the coding region is likely to be more important than when the marker is in non-coding region, right, in other regions. And also there's a lot of studies have been done to accumulate information about the functional relevance of different parts in the genome. So there's, even though the setup so far assume all the markers are independent, are the same, but what I'm trying to say there's a lot of information there people have gained to allow you to really characterize or quantify the functional potential of different parts of the genome. And so have we done also some work. So just give one example to show that. And so this is region hemoglobin gene region. This is if you look across different organs, different tissues, we can actually assign a score of functional relevance in this part of genome. High bar means it's likely to be functional. That's for the whole gene, for organism, the whole human body. But we know there's a difference between brain, between lung, between heart. Actually, we can use the relevant data to estimate the potential functional relevance for different parts of the, for different tissues, right? This brain, so this region is pretty silent. Lung is maybe a little bit more active, blood is more active, right? So now not only do we know these markers are in the genome, also we know their functional relevance. So that actually this information can be utilized in combination with the model I just described to answer some interesting biological questions. For example, we can ask what, for example, schizophrenia, we know it's maybe brain like a disease. We can ask a question of whether the GWAS data tell us so, right? For other disease, we may or may not know the truly the functional or the relevant disease tissue type. These data may tell us. Why? Because if you overlay the GWAS signals with the functional annotations, right? For example, if a disease is a blood related, right? We probably see more overlap between the GWAS signals and the functional regions in the blood, right? So this is all intuitive, but how to really translate that, it'll be a more rigorous way. We can just use the random effects model I just described to you, right? So instead of doing this estimate heritability for the whole genome, we can estimate heritability for the functional part of the genome for given tissue. Because different tissues, they have different functional parts, we can see which tissue the functional part explains the most sort of the phenotype. So this is a very basic idea. And again, it's a very simple model, but we couple with the biological information there, we can really get some interesting results. For example, for height, we can ask which tissue is most relevant for height in terms of genetic, from genetic perspective. It turns out to be muscle and epithelium. And for body mass index, for both males and females, it turned out to be brain. That's more relevant for someone's body mass index. It's not the GI system. On the other hand, for waist-hip ratio, another way to quantify the obesity, for males, it's more muscle. For females, that's more the GI. Okay. And the most interesting part for our research was really to see the Alzheimer disease and Parkinson disease. So these are the two most common forms of neurodegenerative disorders. So there's a problem with your brain, right? And but when you overlay the GWAS signal for Alzheimer disease and Parkinson disease, you see the most enriched signals actually from the immune system. It's not from the brain. And also you can also do that at the cell type level. Okay, so that's very good to know. And also we just show you that this very simple model coupled with annotation data actually can allow us to identify the tissues that are related to a given disease. There are some expected discoveries, also some unexpected discoveries. But now we can actually extend that to look at two traits. So for one trait, we have one linear model to relate the trait to the additive effects of all the SNPs. And now the second trait, we have a second linear model here, right? And so this is the two linear models. And for two traits, this beta I quantifies the genetic effects of the ith marker or ith SNP, ith variant on the first trait. The gamma I quantifies the effect size on the second trait, right? So if the beta I and gamma I, they are independent in the meanwhile, what this SNP or what this variant does to one trait has nothing to do with the other trait. And it corresponds beta and gamma has a 0 correlation, and we can say these two traits they are not genetically correlated. But on the other hand, if beta I and gamma I they are related, right? That means how large effect a SNP has on one trait may be correlated with the effects on the other trait, and this is called genetic correlation here. And we can quantify that genetic correlation by looking at the covariance, and this is genetic covariance. Actually, this phenomenon is called pleiotropy. In genetic, pleio means multiple. It's a one gene or one variant actually can affect on more than one trait. And if you have individual level data, and you can apply the same REML framework to estimate on the covariance here. Again, it seems like this REML estimate in the covariance part still is a very robust to marker dependence. And also the genetic correlation can also be estimated when you only have a summary statistics, not only just for the standard individual level data, we can do REML, but for summary, also we can do LD score, and also we can do other method. This is a paper we published a few years ago. You can estimate that. And also the estimate, this is the efficiency for when you have independent SNPs. Actually, you don't lose much efficiency when the markers are independent. On the other hand, when the markers are dependent, you do lose. You can actually lose quite a bit. But still, you can see these estimates are very robust. And this is what we approved in the paper that's under review to show that for summary stat, I mean, despite this model specified, even when you have some kind of LD structure, as long as you have block independence, the estimates are still unbiased. So we can prove that mathematically that's the case. And when we apply this approach to look at genetic correlations among different traits, so this is about 55 traits studied here, and you can see there's a blocks of traits. You can just by visually they have positive correlations. This here the blue means positive correlation, red means negative correlation, star means a significant, right? So if you see a lot of significant positive genetic correlation among these traits, what these traits are, it's all autoimmune diseases, right? And so these are all having to do with psychiatric disorders. And this has to do with the cognitive function, education. And you can see the negative correlation here is the cognitive function is genetically has a negative genetic correlation with psychiatric disorders. So this model allows us to look at the overall genetic correlation, right? And between different traits. So that's going beyond heritability estimate. But also, just like what we did before, to partition that on the overall heritability into contribution from different tissues. Also, we can partition the overall the covariance into different tissues too. Like for example, between body weight and height, they are genetically correlated. And also we say the genetic correlation mostly come from the brain and also the immune system, right? So this we can based on data we can estimate. Okay, yeah. So that brings me to the last topic I'd like to share with you. So just recap what I have talked about. So I start from this very simple additive model from Fisher in 1918, trying to look at the genetic contribution, quantify genetic contribution to a given trait using relatives. Now with the genetic data, we can actually both identify genes associated with different traits, but also use the genetic data in unrelated individuals to estimate heritability, sort of heritability, right? And also based on same model, if you couple that with the genetic annotation data, right? If you know what regions in the genome are functional in given tissue, we can try to prioritize, identify the tissues most relevant for a given disease. And then we can actually extend a single one model to two models to relate two different traits. Therefore, the genetic correlation between traits. And not only can we really look at the genetic correlation over the genome, but also we can partition that into different tissues. Okay. So this is all good. But now the overall genetic correlation, we can partition into tissues, but also we can really partition into different chromosomal regions, right? So in this paper is under revision, genome biology. They call the SuperGNOVA. So this method is, NOVA is a method to really look at the global genetic correlation. This is a method to look at some local correlation, right? The local correlation, the challenge there is that for the global correlation, we have many markers. For local correlation, we do not have many markers. So we have a reduced amount of information. So even more, the markers are going to be in strong LD dependent with each other. So we have to make some changes, updates of our methods. We have to do some icon decomposition. So I will skip the details here by saying we have to do some modification of our method. But the bottom line, so we are able actually to estimate the genetic correlation between two traits at the local level, not just the whole global level, but more the local level. And so this just gives you the overall estimated genetic correlation, but also tells you the proportion of the genome that are correlated. So this gives you one example to this is to show. Now we can really look at covariance between two traits across all different local regions in the genome. And also we can look at covariance between the two traits using the global markers, right? If we add up the local covariance, it turns out actually they add up pretty much to the global covariance. So that says this is good, right? If you add up the local thing, it's going to be equal to the global thing. But on the other hand, according to which trait pairs you're looking at, the high genetic correlation, global genetic correlation, maybe only capture, may only identify a small proportion of the genome, right? And showing some correlation, whereas a large part of genome may not have much correlation. For example, in this particular case, there's a lot of the genome-wide genetic correlation here. Maybe it's pretty high. This number like Crohn IBD is a pretty high genetic correlation. It's two diseases. But if you look at what proportion of the genome they have significant local correlation, it's like only like above like 12%. So it's only a small proportion of the genome, the correlation, the global contribute, the global, the overall global genetic correlation is only contribute by a small fraction of the genome, right? On the other hand, this is a cognitive performance, education attainment. It's really contribute by many local part, almost like the whole genome actually contribute. So even though for the same level of global contribution, it may have very different local like participations on the other way. Yeah, so the last example here to me is the most interesting part is local genetic correlation. Was this done showed by Yi Liang and Qiong Shi was. This is really a puzzle. That's kind of really that showed why we need to do this local correlation thing. So if you look at autism, right? Autism we know is a genetic disease, and a lot of research has been done to identify things. So there's also autism GWAS. If you look at the global genetic correlation, autism and cognitive performance has a positive correlation. That if you know autism, you may say the autistic kids they tend to have learning disability. How can there's a global positive genetic correlation here between autism and cognitive performance? Not only is a positive correlated with the cognitive function, but also is education attainment. For both measures, measuring the cognitive ability of someone, somehow autism seems to be a good thing. But on the other hand, for another like a disorder called ADHD, attention deficit hyperactive disorder, that's more in line with what you expect to see. It's a negative correlated with cognitive performance and negative correlated with education attainment. But if you look at the genetic correlation between autism and ADHD, they show positive correlation, right? So how to really reconcile this, the seemingly contradictory global genetic correlation patterns here? And I think become a little bit more clear if we really break that down into local correlation, right? If you look at it globally, for example, there may be positive correlation between autism and cognitive performance. But if you look at locally, there are actually a lot of different parts in the genome. Actually, there's a negative correlation between autism and cognitive performance. These regions may be more in line with what we expect to see because autistic kids, they tend to have learning challenges, right? And so if you look at this one, you can just see there's about 27 regions in the genome. There's negative correlation between ADHD and cognitive performance. But also there's region in the genome like ASD, and also there's a number, also depending on how you might make the cut. And so this contradictory result at a global level may be reconciled by actually having different local regions having either positive or negative correlations. And even more, if we can partition the genome into positive regions and negative regions, in the sense that certain regions, they show positive genetic correlation between autism and the cognitive performance. Other regions may show negative. So we can use those regions to come up with a score, or we can come up with a way to predict the cognitive function or predict the risk for autism using the positive regions or negative regions, right? So these are the people, when the kids are partitioned, people are partitioned into different regions, right? This is the into different scores. This is based on the scores derived from SNPs in the negative regions. It's based on the scores for the SNPs in the positive regions. You can see at the beginning there is a much, but look at the tails. Look at the tail, you can see the start of divergence of these two different regions derived PRS. Whereas the negative regions, those people there, they tend to have low IQ, whereas the positive region, they tend to have a high IQ. So this says that this local genetic correlation is a real thing, and also there's another evidence to suggest that the autism actually is a lot of heterogeneity there, and you just use a global genetic correlation. Actually, you may be mislead. You may actually miss some of the interesting patterns. And also, we look at the negative regions and positive regions. They are enriched for different sort of biological processes. Okay. So I'll just say. So today, I basically have been really talking about these two models, right? This is the Fisher's 1918 model, where there's one equation relating the trait to a lot of different genes, and using either relative data from his time or the unrated individuals with the genetic marker data from the current time. Is still the same underlying model, and still this model seems to still actually be providing a very good characterization of the relationship between phenotypes and the genotype, despite its seemingly is a very simple form. And then also I talk about the two models, right? Like model 1, model 2, but still like to think about this model for trait 1, model for trait 2, and talk about the genetic correlation, right, between like a beta and gamma, and actually we learn a lot of interesting the so-called pleiotropy. Also, we can define at the global level. Also, we can look at the local level. Actually, there's some interesting local patterns there we can learn. And again, to summarize, the Fisher's 1918 model, it turned out to provide a rather accurate description on connecting phenotype to phenotype. So there are some kind of little twists, and people have been proposing. And this framework actually has enabled estimation the chip-based heritability for hundreds of, if not thousands, human traits, even the literature. And also it further leads to insights on the genetic architecture of many traits when other information is considered. Yeah, there's much more can be learned from this framework, and Chan and they are working on the causal relationship among different traits, and the whole genome sequence data on 3 billion base pairs, and also the interplay, the interaction between genetic factors and other factors. So far today, I'm only talking about genetic factors, but other factors like the nutrition, the lifestyle, exercise, other things. Yeah, and also you can really track on people over time and to see actually when people age, and the things change. Actually, the heritability for intelligence increases over time, and so this interesting data actually present a lot of challenges in methodology, computation, as well as a theory. So whatever you want to do methodology, you want to do computation, you want to do theory, you can pick and choose. There's a lot of interesting problems here. Again, I really want to acknowledge Chan who's in the audience and who really start to think about this problem along this path, together with Jiming Jiang and Dabeshes, and to have the first paper in the Annotations that to talk about theoretical properties, and more on the methodology end, Qing Shi and Yi Liang, and really have been pushing the NOVA, SuperGNOVA, and Wei Jiang, and to help us to really fill in some of the methodology and also theory gaps. And also I thank the people in my lab. That's how my lab was evolving over time, looking like, and this is how my lab looks like in the COVID times. And also thank the NSF's grant support. So I'll just stop here and open if you have any questions. Thank you.
Thank you, Professor Zhao. So now is open for questions. Please unmute your microphone to ask the question directly, or you put your question on the chat room so that we can also discuss it. Yeah. Any question from the floor? Tijun, you have some question, or yeah? Oh yeah, please. Please.
Yeah. This is Yuan Huang. Thanks, Hongyu, for the very nice lecture, and I quite enjoyed that. I actually have two questions. One is the genetics and the environment interaction. It looks like you did a lot of estimation on the heritability, primarily focused on genetics, and I wonder whether the heritability has any effects on affected by the environment factors, so that in different environment, for example, smoking and non-smoking people, they may have a different, the genetics contribution may be different. And my second question is that you talked about a bit on the prediction, and I wonder, because now most models, it's a linear model, right? And for the prediction, actually sometimes non-linear models between multiple SNPs may work a bit better. I'm not very sure how this in this field, and so I'd like to hear some of your comments.
Okay, thank you, Yuan. Two great questions. The first one is heritability. Really is a not biological thing. It really depends on the environment, right? For example, if you put all the mice in the same cage, look at certain trait, heritability will be higher, right? The environment is very controlled, and if you put these these mice or rats into different environment, there's going to be more heterogeneity. Therefore, the genetic contribution will be lower. So it's not like it also depends on each population you're looking at. So, but it's more give a rough idea. But surprisingly, for height, that's been pretty constant. But if you look people from different cohorts, like people born in the 50s, born in the 90s, then actually probably go see maybe lower heritability, right? Because the environment could change a bit. But in terms of gene environment interaction, and it depends on what scale you are looking at. For example, when we look at the interaction between the genetic risk, for example, for coronary artery disease, and in connection with the lifestyle factors like smoking and exercise, on the hazard level is independent. But if you look at absolute risk, actually there's a dependent. It's really dependent on the skill, the interaction there. It's not like just the same thing as heritability. As our second question about the prediction models, this is surprisingly this additive model has turned out to be pretty good, right? There's a lot of methods have been proposed, including Partition, Hong Kong Yale, and there's a method using the summary step based on lasso, like a lasso sum. It has also based additive model, and that's really going. It's pretty good. And some people in my lab, and they try to use some like deep learning tools to really see whether that can give you a better prediction. Couldn't, right? Because maybe there's a couple of things. One is the signals are very weak here, and it's not talking about the images. So it's a very different game. The second one is biologically speaking, maybe at the molecular level, there's a lot of interactions, but at the phenotype level are going to be many steps over, and the additive model actually provides pretty good fit there. I see someone raising hand, right? Yeah.
Thank you.
Okay.
Yeah,
thank you, Dr. Wang. So in the chat room, there's one question asking about the immune system is more related to the AD and PD than the brain. Professor, could you elaborate a little more on that?
Yeah, I think in the Alzheimer field, in the last 5 or 10 years, people start to realize the local immune environment is very important. Actually, it turns out to be the microglia. It's really the cell type that plays a very important role for the onset of Alzheimer's disease. Yeah, and also when we look at the single cell data, also the bulk RNA data from the Alzheimer brain patients, we do see there's a clear signature, the immune signature for the look at the difference between Alzheimer brains and then sort of a healthy postmortem brains. And for the single cell level data, we can look at the cell type specific the expressions. And the most signals actually we were able to observe actually at microglia cells, both for Alzheimer as well for Parkinson. So that seemed to be suggesting that really is I think cooperation between both the genetic studies as well as genomic studies implicating the importance of the immune system for Alzheimer's disease, as well for Parkinson's disease.
Okay. Another question at the chat room is about the random effect model. Why would we assume the effects in the same trait are independent if we already know that there exists LD between the SNPs?
That's a great question again, but I think we have to make some assumption. That's probably the markers are, when the markers are dependent on each other, that I think that doesn't, one marker being important biologically doesn't diminish the chance the other marker also being biologically important. So in that way, we want to treat all the marker equally in that sense, right? I mean, that's, you can think about other ways. And there's other models. This is one random effects model. There's other model, LDAK model, and that's from the David Balding and the Doug Speed. They have been writing papers in Nature Genetics, and they're trying to promote their model. So there's somewhat different setup where they try to assign markers in different LDs, the different LD strands with a different chance or different opportunity to play a more or less important role in the genetic determinants of the traits. So there are alternative models there, I'm saying, and some people argue those models may give a little bit better fit to the data, but I think the difference so far to me has been pretty minuscule. Yeah.
Yeah, but in the modeling, can we incorporate like the LD information in the model?
I think the LD score regression is really explicit model LD in the model. It's in the model already. Yeah, but I think the practical limitation is that when you only have a summary statistics, you do not really observe the true LD patterns for the samples you have. You have to use a reference panel. Yeah, if there's a disconnect between what you have and the reference panel, there may be some bias. So that's been a challenging problem. But if you have individual level data and just go straight using the individual level data, actually, in the Chan knows this much more than I do, how to manage that computationally. That's a very challenging thing to do, managing a large data set with 500,000 people. That's not trivial. Yeah.
Yeah, I remember I did a work with Pat Sham, is to incorporate the LD information in the selection of the tagging SNP. But I think that is the different thing about this kind of random effect model.
Absolutely, it's a different, so it's a different concept. I'm trying to more,
Okay. Another question at the chat room is about the interpretation on and application of partitioning on genetic correlation. If we know, for example, the height and BMI genetic correlation can be partitioned to some tissues, what does it mean? How could we further make use of the conclusions? I'm not so sure I understand the question fully, but how about
So for example, like maybe let's go, maybe the simple example would be if you partition the genome into like 22 autosomes and the sex chromosome, right? Yeah, so instead of calculating covariance between two traits using all the markers in the genome, you can calculate the covariance between the traits using the markers in on chromosome 1, chromosome 2, chromosome 3, right? Yeah, you can really see which chromosome contributes most to the covariance. Yeah, that's also the figure I showed toward the end, the local genetic correlation or local genetic covariance. And really depends on the traits. For example, for the between cognitive performance and educational attainment, all the different regions they contribute, right? On the other hand, for example, between Alzheimer, between autism and cognitive, there may be certain regions contribute more than others, and we can really see what those regions are, and then give you some insights about. But back to the question about the different tissues. Yeah, suppose in the ideal world, there's non-overlapping tissue annotations in the genome. We can partition that into different things and to see which tissue, the function of which, because each tissue has different functional regions, right? We can look at which tissue's functional regions contribute most to the covariance. And that gives an idea this tissue may be relevant, maybe contribute most to the observed covariance between different traits.
Okay.
Hi.
Oh, Pat. Okay.
Hi, Pat. I didn't know you're here.
Thank you very much for your great talk.
Yeah. Thank you, Pat.
Yes, I have a question about the last part, which is the supernova. So
yeah.
In terms of the local contributions, how small can local be? What is the sort of the smallest sort of genomic units that you think you can sort of reliably sort of estimate the local contribution? I suppose,
yeah,
to follow on from that, what do you think are the sort of determination? Is there are you looking into how you can predict, these different patterns? Say, the regions where the local contribution is say greater than others, or have different correlation. Is there any way you can sort of predict that by looking at the genes that are contained, or some sort of differences in annotation?
Right. And so first, there's maybe three parts. One is just a question about how many local regions we are looking at. It's somewhere between like 1,500 regions we partition the whole genome into. And how that can be utilized. So one thing is we can really, for example, on the autism and the cognitive performance, we can really look at what regions show positive or negative, significant positive, significant negative covariance or correlation there, and that give us some hint what may be. The paper is on the by archive. I can email update version. So that one we are looking at sort of like the positive region, negative regions. They seem to be involved in different biological process of annotation. But and also we can there's people have been using MTAG, right? We look at the borrowed correlated traits, and also we can have some corresponding version called local MTAG, right? So we can really look at the so we have been applying to disease like inflammatory, there's an idiopathic pulmonary fibrosis, also COPD. For example, you can look at, look at local region to see what traits are significantly correlated in the local region for that particular target trait, and using the MTAG idea of a more local version and to borrow information from other traits to improve. So for a lot of traits, we have an example to show there's no overall genetic correlation at the global level. But locally, actually, there's significant correlation. We can really borrow information from those traits to help us to improve the signal for the target trait.
Yes, that's very interesting.
Yeah.
I mean, even if the genetic correlation is 0, there can be pleiotropy.
Absolutely, yeah.
And
locally,
it's just a positive and a negative.
In theory, they may cancel out, right? Yeah, we do have those. Yeah.
I think that's particularly interesting when there are sort of multiple tissue types that are involved in disease, because even though there may be one predominant tissue like brain for body weight, but there are also there are other tissue types that are relevant. So it could be that.
And also we can also get down all the way down to the cell type level. Yeah.
Great. Thank you very much.
All right. Thank you, Pat.
Interested in your your recent paper.
Thank you.
And forward that.
Okay. Yeah, I will. Yeah, I will send those to you. Yeah.
So any question from the phone? Okay. And also now no more question from the Zoom. Yeah, maybe. Yeah. Sorry.
Is there one more at the chat?
Oh, more in the chat box. Oh yeah. I just saw. Yeah. So if this question comes up and it's still there are obviously still a lot of factors that affect the prediction of the heritability, such as not enough data, epigenetics, feed from the public, and so on. How are the researchers now? What can we do in the future to improve the methodology? These are open questions. Talking about the limitation or something development.
Yeah, this is I think the not enough data. If we believe in the model, then it seemed to be like we will have pretty unbiased estimate, but I think that the variance part maybe will reduce as we get more and more data. But I think for the sample size that we are dealing with for the most traits, if we believe in the model, the heritability estimate, I think Peg and Tan can comment, stabilizing in a way, right? For a lot of the common traits, we roughly know it just seems to be, at least for the different European population, different stock cohorts, it's pretty stable even across populations. Epigenetic. So the epigenetic, if you think more as an environmental factor of fighting the epigenetics patterns, then that may not be part of the genetic contribution. But on the other hand, for the genetic control of the epigenetic patterns, and if you think that the SNPs they contribute to the epigenetic machinery, that may capture certain part of, because epigenetics can be a lot of different things, right? Can be methylation, can be histone-modification. And for the methylation, that can be maybe under the methylation QTL and also maybe under environment. So it's a mixed bag to me. And I really want to get down to what this is. A fear from the public. That might maybe have to do with, I assume, I'm trying to interpret that, may have to do with whether the people answer the questions right or wrong, right? For if you ask certain sensitive questions, people may not be giving the right answer, like for example, income, right? And other things people make, and maybe other questions maybe tend to exaggerate or tend to diminish what they reduce what they have. So I think that I think this has to do with the measurement, the instrument, right? Hopefully we can get a more reliable instrument. But for a lot of these, I mean, even for the electronic medical records, right? A lot of people, they're making a living just to derive phenotypes from electronic medical, realizing the limitation there. So there's going to be, I mean, it's a great question about the phenotype. Even people are truthful, they may not be really be able to recall or tell you the true phenotype just because like one reason or the other.
I think definitely the both the machine.
Oh, these are all great questions. Yeah.
And then to get more and more data. But of course, the privacy is another issue as a long one, especially talking about human the personal data. But in any case, you also develop some summary statistics to help to solve some of the issues.
Yeah. So one time I saw there, I think if you read the original the report, there's an Obama precision medicine program. Now it's called All for Us. In the original report, actually, the people did some careful power analysis to justify why they need 1 million people, right? Just because you look at all different phenotypes, you break it down, and 1 million people will give you a pretty good estimate, a lot of different things for a lot of common traits. I mean, that if you go below that, you may or may not, but this sort of, to me, that's really fascinating to read that part when it first came out. Maybe now, looking back 6 or 7 years ago, I think that's very interesting. I mean, maybe 4 or 5 years ago. I mean, that before Trump took over the office, that right after. Well, Obama's going to step down or face off. Yeah, so that report is very interesting. Yeah, how they designed the study. Yeah.
Okay. So I don't. Any other questions from the floor or from the chat room? If no more, then because of the come to the end of the distinguished lecture. So let's thank Professor ZHAO again for his insightful distinguished lecture and introduction the whole development of this genetic model. Yeah. So thanks all the participation again. We look forward to you joining the next distinguished lecture by the Faculty of Science. Okay. Thank you very much. Thank you, Professor.